தமிழ் கோர்வை சேனல் சார்பாக அனைவருக்கும் வணக்கம். என் பெயர் கண்ணதாசன். Today we are going to discuss about structure of DNA part 1. The expansion for DNA here is deoxyribonucleic acid. Okay, first let us understand about the amazing facts of DNA. Amazing facts of DNA. You see, still as a human being we are carrying few DNA first formed in the bacteria. Not only in our body, these few segments are present in the higher plants also, other animals as well as microorganisms. Even we are able to establish the link with our ancestors, fossilized ancestors, maybe with the fossils obtained at various levels. So one day it is possible, with the help of nucleic acids, we are able to trace our ancestral bacteria also. So let us start with the elements of DNA. So this is the actual subject. What are the elements which construct the DNA and the DNA do all these wonders in the world? There are 5 elements only, right? They are carbon, hydrogen, oxygen, nitrogen and phosphate. These are the 5 elements. These 5 elements in various combinations produce 3 important groups of molecules. They are nitrogen bases, deoxyribose sugar, phosphoric acid. First, let us take the nitrogen bases. This again consists of 2 broad categories. One is purines. It consists of adenine and guanine. These are also nitrogen bases coming under purines. Then pyrimidines, cytosine and thymine, and their symbols are also given here as AgCT. First, let us discuss about purines. It is a double carbon and nitrogen ring. Heterocyclic aromatic organic compounds. Heterocyclic aromatic organic compound. Okay, and it consists of 4 nitrogen atoms. You see, this is one ring, this is another ring. 2 rings are present, and 4 nitrogen atoms are present in adenine and guanine. 4 nitrogen atoms. Okay, this position, you please draw the diagram, practice it, and mark this position. Questions can be in the competitive examination like NEET asked based on these numbers also. Okay, what are the groups present in these positions? Next, pyrimidines. Single ring structure consists of one hydrogen carbon ring, only one ring with 2 nitrogen atoms. You see here 2 nitrogen atoms in cytosine, here 2 nitrogen atoms in thymine. Next, the difference between purines and pyrimidines. Here, you see, purine consists of one pyrimidine ring and one imidazole ring, whereas pyrimidine consists of only one ring, as we have already discussed. The next, let us go to the melting point. Here it is 214 degrees Celsius. In one of my reference, it is given. The melting point in pyrimidine is just 22 degrees Celsius here. Then catabolism, if the breakdown occurs within the cell, okay, the uric acid is produced as a waste product, and here if the breakdown occurs in pyrimidine, it produces carbon dioxide, beta amino acid and ammonia. Next molecule is deoxyribose sugar. I compared it with the ribose sugar. Both the sugar made up of 5 carbons and their positions are given here 1 to 5, and the second carbon, oxygen and hydrogen, OH group is present in ribose, but oxygen is absent in deoxyribose, so it is called as deoxyribose. Okay, third molecule is phosphoric acid, H3PO4. It contribute for the formation of ester bond. Okay, this oxygen establish ester bond with the previous carbon. Okay, 3 dash carbon. Okay, and also this OH establish ester bond with the following ribose sugar. Okay, so by removal of the hydrogen, so as it yield hydrogen, it become acid. DNA also become acid, and also as the protons are going out of the molecule, the DNA also become negatively charged molecule. Okay, so this is the reason why the DNA is negatively charged. Now, naming of DNA. As we have discussed these molecules, now it is the time to discuss why the name is given like this. Deoxyribose sugar is present, so D stand for deoxyribose sugar. It was first discovered in the nucleus, so it is nucleic. It is acid in nature because of the presence of phosphoric acid, so it is acid. So deoxyribonucleic acid. It is negatively charged, as I already told you. Okay, because of the presence of oxygen atom. Okay, hydrogen also removed. Okay, as the hydrogen atom are removed, okay, oxygen provides the negative charge. Okay, so the DNA molecule is negatively charged molecule, and the molecules, elements in combination produce molecules. Molecule in combination first produce nucleoside. You see, this is the purine. Okay, the ninth nitrogen with the deoxyribose sugar, first carbon, produce glycosidic bond. Now this is called as nucleoside. The same way, pyrimidine also, first nitrogen of a pyrimidine and first carbon of the deoxyribose sugar establish glycosidic bond and produce one nucleoside. Here, nucleotide. This is the monomer of the polymer DNA. Okay, so nucleoside plus one phosphate group is called as nucleotide. So here I have given nucleotide of thymine. Okay, all the three molecules are present, and nucleotide of cytosine, guanine, and adenine. Now polymerization. Polymerization of the nucleotide occur by the condensation reaction. Okay, here the condensation reaction is given phosphodiester bond. This is the backbone of the DNA. You see, this is the first nucleotide. This is the second nucleotide. The first nucleotide 3 dash oxygen. Okay, 3 dash carbon establish ester bond with oxygen of phosphate. In the same way, fifth carbon of following deoxyribose sugar establish ester bond with the oxygen of the phosphate. So two ester bonds are produced from one phosphate. Here it is called as phosphodiester bond. Likewise, continuously phosphodiester bonds are formed. So it becomes the one DNA strand. So it is the backbone of DNA. Polymerization of nucleotide. The condensation reaction I told you. This OH group of phosphate and this hydrogen of previous deoxyribose sugar join together and form H2O. Phosphodiester bonds are formed. Polarity also established in the DNA strand because of this. You see here, this is the DNA strand, polymerized DNA strand. Here it consists of 5 dash carbon, it is free here. In this 3 dash carbon is free. So the polarity is said to be here 5 dash to 3 dash direction. 5 dash to 3 dash direction. Okay, next one is nucleotide polymerization reaction. Phosphodiester bonds are already formed, you see, between two nucleotides. And here this is the new nucleotide is coming. Always the nucleotide, free nucleotide in the cell present in the form of, say example, adenine means adenosine triphosphate. With the 3-phosphate only it is present. Very clear. So in the same way, guanosine triphosphate, cytosine triphosphate, likewise they are present. Once they are coming to the polymerization reaction, once they are coming to the formation of ester bond, these 2 phosphates are removed. You see, and ester bond is formed. Are you clear? So the phosphates are removed. Point number 1. Water molecule also removed. That is point number 2. These are the 2 points you should remember during the polymerization reaction and also formation of phosphodiester bond. Okay, this is the end of part 1, students. Thank you for watching. If you like this video, press the like button, share it, subscribe our channel, and press the bell icon to receive our notification. See you in the next video, DNA part 2. Thank you, students.